Have you ever wondered what causes genetic disorders like Down syndrome? Well, the answer lies in a phenomenon known as aneuploidy. Aneuploidy is the presence of an abnormal number of chromosomes in a cell, a condition resulting from errors during cell division, which leads to an unequal distribution of genetic material to the offspring cells. This phenomenon was first discovered over a century ago and has since been linked to a multitude of genetic disorders and even cancer. But it's not just about the number of chromosomes. Aneuploidy can cause a ripple effect, altering the very fabric of cellular function and even contributing to the diversity and evolution of cancerous tumors. It's an intricate dance of genetic material, one that we're only starting to fully understand. Now that we understand what aneuploidy is, let's delve deeper into how it is formed. So how is aneuploidy formed? A seemingly simple question, but the answer is steeped in the intricate dance of cell division and genetic regulation. Let's dive into the mechanisms that contribute to aneuploidy formation. Picture a cell on the brink of division, its chromosomes lined up ready to split evenly into two daughter cells. This process is tightly regulated by the spindle assembly checkpoint, or SAC for short. SAC acts as the gatekeeper, ensuring that chromosomes segregate properly. But imagine if something goes wrong with SAC components, like MAD2 or BABR1. This could lead to a premature separation of chromosomes, resulting in an unequal distribution of genetic material. One cell might end up with more chromosomes than it should, a condition we refer to as aneuploidy. Now, let's spin the focus to the centrosomes, the structures that help in organizing microtubules and directing the separation of chromosomes during cell division. Any perturbations in centrosome duplication and function can throw a wrench in the works of chromosome segregation. If the centrosomes aren't operating as they should, chromosomes could misaggregate, giving rise to aneuploidy. Furthermore, DNA repair pathways play a significant role in maintaining our genetic integrity. They act as the body's genetic proofreaders, fixing any errors that occur during DNA replication. But what if these proofreaders make mistakes or miss errors? This could lead to chromosomal aberrations, which could predispose cells to aneuploidy. The usual suspects for these defects include pathways like homologous recombination and non-homologous end joining. So there you have it. Aneuploidy can arise from defects in the spindle assembly checkpoint, perturbations in centrosome duplication and function, and defects in DNA repair pathways. These mechanisms contribute to the formation of aneuploidy. But what happens to the cell once it has an abnormal number of chromosomes? Well, that's a story for the next part of our discussion. Stay tuned. Aneuploidy isn't just a silent spectator inside the cell. It's like an uninvited guest that can turn the entire household upside down. The presence of an abnormal number of chromosomes in a cell can cause significant changes in cellular physiology, including alterations in gene expression, cell proliferation, and programmed cell death or apoptosis. Imagine the chromosomes as the blueprint of a house. If you have an extra copy of the blueprint or are missing one, you might end up with an extra room or a missing wall. Similarly, when cells have an extra or missing chromosome, it can lead to gene dosage imbalances, causing changes in protein expression levels and altering the cell's normal functioning and phenotype. But the effects of aneuploidy don't stop there. It can also trigger stress responses within the cell, like the activation of the p53 pathway, a key player in preventing cancer. This pathway, when activated by aneuploidy, can lead to cell cycle arrest, DNA repair, or apoptosis, thereby affecting cell viability. Additionally, aneuploidy can increase oxidative stress within the cell. Oxidative stress, in simple terms, is an imbalance between the production of free radicals and the body's ability to counteract their harmful effects. This can cause damage to the cell's components, including DNA, proteins, and lipids, and contribute to the pathogenesis of diseases. But perhaps one of the most profound impacts of aneuploidy is its role in promoting genomic instability. This instability facilitates the acquisition of additional mutations, driving the progression of diseases like cancer. It's like a snowball effect. Once started, it's hard to stop. Aneuploidy can also contribute to therapeutic resistance in cancer. Because cancer cells often have abnormal numbers of chromosomes, they can adapt to different conditions and resist treatments, making them more challenging to eradicate. The consequences of aneuploidy are far-reaching and can contribute to disease pathogenesis. But how can we use this knowledge for therapeutic purposes? Stay tuned as we delve deeper into the therapeutic implications of aneuploidy in our next scene. Understanding aneuploidy doesn't just help us understand disease, it also opens up new avenues for treatment. This insight into aneuploidy is like stepping into a new world of therapeutic possibilities. Imagine being able to target key regulators of chromosome segregation for treatment. These regulators include the components of the spindle assembly checkpoint, or SAC, and proteins involved in centrosome functions. By targeting these, we might be able to selectively eliminate aneuploid cells while leaving healthy cells untouched. It's like having a sniper scope pointed directly at the enemy, ensuring minimal collateral damage. But our strategic arsenal doesn't end there. Aneuploidy brings with it certain vulnerabilities. You see, aneuploidy-induced stress responses, like the activation of the p53 pathway and increased oxidative stress, can be exploited. By understanding these responses, we can manipulate them to our advantage. This approach is akin to finding an enemy's weak spot and striking right there, a concept known as synthetic lethality interactions. And let's not forget the exciting advances in genome editing technologies. Tools like CRISPR-Cas9 are not just a part of some science fiction fantasy. They are a reality, offering us the opportunity to correct aneuploidies and restore chromosomal stability in genetic disorders. It's like having a time machine that allows us to go back and fix the errors of the past. These therapeutic implications of aneuploidy hold promise for the future of treating genetic disorders and cancer. And with every new discovery, we get one step closer to making this future a reality. Aneuploidy, a complex yet fascinating aspect of genetics. We've journeyed through its intricate mechanisms, seen its profound impact on cellular function, and explored its therapeutic implications. From the spindle assembly checkpoint to genome editing technologies, understanding aneuploidy brings us closer to targeted therapies that can exploit chromosomal abnormalities. But our voyage doesn't end here. As we continue to unravel the mysteries of aneuploidy, we inch closer to developing effective therapies for genetic disorders and cancer. Until then, the journey of discovery continues.